Photoperiodism is defined as the response of an organism to seasonal changes in day length. Seasonal changes can be perceived in terms of variation in the length of the day and temperature. Days are longer in summer and hotter. Days are shorter in winter and cooler. Photoperiodism is the physiological response of the organisms to the length of day or night. We will see in this module how do plants perceive the changes in daylight and temperature and respond to different seasons. We have seasonal flowers, fruits and vegetables. In India, during summer we get fruits like mango, watermelon, muskmelon, oranges, etc. and vegetables like okra, cucumber, pumpkin and also flowers like sunflower, marigold and petunia. During winter, we get vegetables like pea, carrot, fenugreek leaves and flowers like chrysanthemum, daisy, rose, etc. How do these plants produce flowers and fruits only in one of the seasons? In other words, how do they know it is summer or winter? Plants can measure day length through the photoreceptors like Phytochrome and Cryptochrome. They use these photoreceptors to sense the seasonal changes in daylight and send signals which bring about different developmental processes. A summer flowering plant will send the signal for development of flower only when the days are longer. Similarly, a winter flowering plant blossoms only when the plant senses shorter days. Besides day length, some temperate plants also need to pass through a cold climate of winter before they can flower. This process is called vernalization and can also be induced artificially by treating the plants with low temperature. The external signals brought about by changes in daylight and temperature induces developmental changes through a complex biochemical reaction involving hormones and genes. Let us understand this interaction of external and internal factors that control seasonal flowering and fruiting in plants. Of light in longer days, more will be photosynthesis. Plant physiologists believed for a long time that flowering was a consequence of the accumulation of photosynthetic products synthesized during long days. This concept was changed by an experiment done by Garner and Allard in 1920. They grew a mutant variety of tobacco, Maryland mammoth, in summer. This variety grew profusely robust during the long days of summer with a high rate of photosynthesis, but failed to produce flowers. But the same variety produced flowers when it was grown during shorter days of winter under natural light when the rate of photosynthesis was lesser. This observation led Garner and Allard to test the effect of artificially provided short days. The artificial short days were provided during the long days of summer by covering the plants late in the afternoon with tents impermeable to light. In this experiment, plants flowered even in the long days of summer. This result disproved the concept that longer periods of sunlight resulting in increase in photosynthesis promote flowering in general. It also confirmed that length of the day or photoperiod was the determining factor in flowering. In this experiment, tobacco proved to be a short day plant which can flower only when the day length is shortened. On the basis of photoperiodic responses, two main categories of plants are recognized: short day plants that flower only in short days and long day plants that flower only in long days. There are certain plants insensitive to day length and they are referred to as day neutral plants. The short day plants are further categorized as qualitative short day plants and quantitative short day plants. The qualitative short day plants do not form flowers in any other season, whereas in the quantitative short day plants, the flowering is accelerated by short days. Similarly, there are qualitative and quantitative long day plants. The former flowers only in long days, and in the later, flowering is accelerated by long days. The day length and the seasons vary in different parts of the earth depending on the latitude. At the equator, day length and night length are equal and constant throughout the year. As we move away from the equator towards the poles, the days become longer in summer and shorter in winter. Depending on the latitude from which they have originated, plant species have evolved specific photoperiodic responses that enables them to detect the seasonal changes in day length. Since plants can measure day length, their specific photoperiod responses are strongly influenced by the latitude in which they grow. In the northern hemisphere, the long day plants typically flower during the spring or early summer as the days are getting longer. The longest day of the year in the northern hemisphere is on or about 21st June. After that date, days grow shorter, that is, nights grow longer until 21st December. The situation is reversed in the southern hemisphere, where the longest day is 21st December and the shortest day is 21st June. India, a tropical country, comes under 10 to 23 degree north, and the day length varies from 12.5 to 13.5 hours in summer and 10.5 to 11 hours in winter. But in a temperate country like Russia, that is in the zone of 50 to 60 degree north latitude, the day length is as short as 7 hours in winter and as long as 16 hours in summer. Days lengthen during spring or early summer, and long day plants can effectively measure this and delay flowering until the critical day length is reached. Many varieties of wheat behave in this way. Similarly, short day plants measure day length when they are shortened below the critical day length, and they often flower in fall. Many varieties of Chrysanthemum morifolium flower when the days are shortened. Phytochrome is the primary photoreceptor in plants involved in measuring the light and dark period in a 24-hour cycle. This is best understood by night break experiments. A night break experiment is interruption of the dark period in a 24-hour cycle of light and dark by a brief exposure to light. A short day plant like Xanthium flowers in a cycle of 8 hours of light and 16 hours of dark. If the dark period is interrupted in the middle by a brief exposure to red light, then flowering is inhibited. The effect of red light is reversible with subsequent exposure to far-red light. The action spectra show that red light is most effective in inhibiting flowering, and this inhibition is reversed maximally at 725 nanometer, that is far-red light. The peak of inhibition and its reversal implies the involvement of phytochrome. Sunlight is richer in red than far-red light. So, at the sundown, all the phytochrome remains as PFR. During night, PFR converts back to PR. Xanthium needs 8.5 hours to convert all PFR to PR. In Xanthium, a short day plant, PR form is needed for the release of the flowering signal. If this process is interrupted by a flash of red light, the PR is immediately reconverted to PFR and the flowering signal is not released. If the exposure to red light is immediately followed by far red, then the PFR is converted back to PR and the flowering takes place. A similar night break experiment with a long day plant shows that red light promotes flowering. This promotion is reversible with subsequent exposure to far red light. Although phytochrome has been shown to be the primary photoreceptor for photoperiodism in both short day and long day plants, recent experiments in Arabidopsis have provided evidence for the involvement of a blue light photoreceptor. The involvement of blue light receptor was demonstrated in Arabidopsis by using H5 mutant, which are deficient in phytochrome. Blue light could promote flowering in these mutants, although they were not able to respond to red and far red light. The photoperiodic stimulus in both short day and long day plants is perceived by the leaves. This can be experimentally proved in a short day plant like Xanthium by treating a single leaf with short photoperiods and keeping the rest of the leaves under long photoperiods. Treatment of a single leaf is sufficient to induce flowering in Xanthium. The process that occurs in leaves, resulting in the transmission of a floral stimulus to the shoot apex, is referred to as photoperiodic induction. The floral stimulus is transported through phloem. The movement of floral signal can be prevented by blocking the phloem transport. Genes have been identified that play a crucial role in the formation of floral organs in response to floral stimulus. Circadian rhythms are the rhythms in biological activities that fluctuate over a period of approximately 24 hours. The word is derived from Latin word circa, meaning about, and dies, meaning day. This rhythm is independent of day and night cycle, which means even under constant environmental conditions like continuous darkness or continuous light, the cycle of activity is maintained. Under constant conditions, the cycle may drift out of phase with the environment. For example, the sleep cycle in humans is adjusted to day and night cycle. When a person travels across the continents, it gets out of phase with the environment, referred to as jet lag. Plants have an innate circadian rhythm that regulates the expression of many genes, including those which control development of flowers. Photoperiod works in combination with circadian rhythm in regulating flowering. This has been best understood in Arabidopsis. Arabidopsis has a gene constans that produces a mRNA whose level rise and fall with circadian rhythm. This mRNA is abundant early in the morning, declines during the middle part of the day, and then rises to another peak late in the afternoon. The constans protein coded by this mRNA is quickly degraded during the morning and the middle part of the day. This degradation is mediated by Phytochrome B. Production of mRNA and protein increases in the late afternoon when the days are longer, mediated by Phytochrome A and cryptochrome. The constans protein turns on flowering locus T gene, which is needed to start the conversion of the apical buds to floral buds in Arabidopsis. In short days, when the sun dips early, darkness falls before the rise in constans mRNA, and hence not enough constans protein is synthesized to induce flowering. So, flowering in Arabidopsis seems to require the interaction of daylight perceived by phytochromes and cryptochromes and the intrinsic circadian rhythm of constans protein. In contrast to the long day plants, constans protein act as inhibitors of flowering in short day plants when their synthesis is enhanced by long days, as in case of rice plants. In some of the trees like populus, the photoperiodism not only controls flowering, but also stops growth of vegetative buds during short days of winter. This is also mediated by constans and flowering locus T genes. Flowering is not only regulated by photoperiod and circadian rhythms, but also by temperature, particularly in temperate plants. Vernalization is induction of plant flowering process by exposure to prolonged cold of winter or by an artificial treatment with low temperature. Vernalization process was first discovered by the Russian scientist Lysenko in 1928. In Russia, farmers knew two types of cereals: the winter cereals, which required chilling for emergence and growth, and summer cereals, which could be sown in spring without passing through a cold winter. Lysenko found a method to treat the winter wheat seeds artificially with low temperature, which made them behave like spring seeds. The word vernalization is a translation of the Russian word jarovization. Spring cereals are called jarov in Russia, and this word was coined by Lysenko. Many plants grown in temperate climates require vernalization and must experience a period of low winter temperature to initiate or accelerate the flowering process. This ensures that reproductive development and seed production occurs in spring and summer rather than in autumn. Typical vernalization temperatures are below 5 to 10 degree centigrade. In nature, Plants requiring vernalization need short winter days for cold treatment, but being long day plants, they also require long day lengths available in spring and summer. Winter annuals like calendula, petunia, pansy, verbena, etc., which are sown in the fall and flower in the following summer, respond to low temperature very early in their life cycle. Biennials like carrot, onion, hollyhock, etc., grow as rosettes in the first season and flower in the next season. In winter annual ecotypes of Arabidopsis that require both vernalization and long days to flower, a gene, flowering locus C, that acts as a repressor of flowering has been identified. This gene is highly expressed in a non-vernalized shoot apical meristem, but its expression is reduced by vernalization or exposure to cold, which permits flowering. Seasonally produce flowers and fruits in response to external factors. Day length and temperature play a crucial role in the production of flowers through photoperiod and vernalization. Genes have been identified that play a crucial role in the formation of floral organs in response to both endogenous factors like circadian rhythms and hormones, and the external factors like photoperiod and temperature.